Life just simply isn't possible without energy. It is vital for human functions such as growth and repair, respiration, and blood transport. Larger amounts of energy are needed for physical tasks including work, exercise, and recreational activities such as sports. Energy is derived from the food we eat. Carbohydrates and fats are the macronutrients that supply the most energy per gram, with
fats consist of a glycerol molecule bonded to 3 fatty acid chains. In tissues with high energy requirements, such as the heart, up to 50 to 70% of energy in the form of ATP production comes from fatty acid beta-oxidation. In eukaryotic cells, beta-oxidation takes place in the mitochondria and peroxisomes. Let's have a closer look at this process in a eukaryotic cell.
For beta-oxidation to take place, fatty acids must first enter the cell through the cell membrane, then bind to coenzyme A, forming acyl-CoA. First, fatty acid protein transporters allow fatty acids to cross the cell membrane, as the negatively charged fatty acid chains cannot cross it alone. The enzyme fatty acyl-CoA synthase adds a CoA group to the fatty acid chain, converting it to acyl-CoA. There are then 2 ways for acyl-CoA to enter the mitochondria. If the acyl-CoA chain is too short, it can freely diffuse through the mitochondrial membrane. However, if it is too long, enzyme carnitine palmitoyl transferase 1 converts acyl-CoA to an acyl-carnitine chain, which can be transported across the mitochondrial membrane by carnitine translocase. Once there, it reverts back to acyl-CoA via the enzyme carnitine palmitoyl transferase 2. This process is the carnitine shuttle, which happens in the peroxisomes. Once inside the mitochondria, beta-oxidation can finally begin.
There are 4 stages to beta-oxidation, which consist of dehydrogenation, hydration, oxidation, and photolysis. In dehydrogenation, acyl-CoA is oxidized by the enzyme acyl-CoA dehydrogenase. A double bond is formed between the second and third carbons of the acyl-CoA chain. The end product of this reaction is trans-delta squared enoyl-CoA. The reaction also uses FAD and produces FADH2, which enters the citric cycle to produce ATP to be used as energy.
Hydration, which is the second step. The double bonds between C2 and C3 of trans-delta squared enoyl-CoA is hydrated. Of course, water is needed, forming the end product L-beta hydroxyacyl-CoA, which has a hydroxyl group of C2 in place of the double bond. This reaction is catalyzed by another enzyme called enoyl-CoA hydrase.
The third stage is oxidation. The hydroxyl group in carbon 2 of L-beta hydroxyacyl coenzyme A is oxidized by NAD+ in a reaction that is catalyzed by the enzyme 3-hydroxyacyl coenzyme A dehydrogenase, producing beta-ketoacyl coenzyme A and NADH+H. NADH can now enter the citric acid cycle to produce ATP that will be used as energy.
The last stage is thiolysis. Beta-ketoacyl CoA is cleaved by a thiol group of another CoA molecule. The cleavage takes place between C2 and C3. Therefore, the end products are an acetyl CoA molecule with the original first carbons, C1 and C2, and an acyl CoA chain 2 carbons shorter than the original acyl CoA chain that entered the beta-oxidation cycle.
To summarize, beta-oxidation is a catabolic process that converts fatty acid molecules into acetyl-CoA. As each round of beta-oxidation produces 2-carbon acetyl-CoA, it takes several rounds to completely break down a fatty acid. Given this, it is useful to think of beta-oxidation as a cycle. The cycle continues until a 4 or 5 carbon fatty acid acetyl-CoA remains.
This process occurs during a fasted state when the body needs energy. When you are full, the body would much rather store triglyceride molecules as a deposed tissue.